Hello, everyone. So for this research proposal, we are to evaluate the impacts of sedimentation on the sea cucumber Cycropodes discreta in the Clarion-Clipperton zone. This region, the CCZ Clarion-Clipperton zone, was selected due to prospective deep ocean mining that may occur here in the near future and the conservation implications of that from the species perspective. The research will be done mainly from video and time-lapse footage, which you can see here for another member of the same genus, the Cycropodes sea cucumber genus. Our research questions for the study are as follows. One, how does suspended sedimentation impact the feeding of P. discreta? And how does this sedimentation impact the movement of the species, such as with distance covered as well? And sedimentation here in this case is again within the context of deep ocean mining, potential sediment plumes that may result from said mining. Our hypotheses are as follows. One, for null hypothesis, sedimentation will not have a significant effect on the feeding or the movement of P. discreta. Whereas one alternative hypothesis is that if you have higher suspended sedimentation rates, then feeding rates and total movement will decrease from that. The basis of this alternative hypothesis is from Stenbers et al. 2023, which did a laboratory study on sediment responses from the deep sea jelly Paraphylla. This graph at the top shows sediment concentration on the x-axis and health score on the y-axis. And as you can see, with higher sediment concentration and over time, over a period of 24 hours, as indicated by these color bars, 24 hours, the darkest bar, health scores are going down in many of these cases. Likewise, this diagram illustrates an observed response from the jellies to higher sediment concentrations, and that is the production of mucus, which can be a sign of an immunity response, which likewise may be detrimental to their overall health. Now, for the research design itself, we will use simple random sampling to identify the cucumbers to be used in the study. Samples will be enclosed within a 5 by 5 meter grid to conduct about 2-hour trials, 3 2-hour trials each day. Random assignment will be a control group of no sedimentation and a treatment group of 25 milligrams per liter sediment. That's an intermediate of some of the treatments in Stenbers et al. We'll use equipment such as ROVs, remotely operated vehicles, which can handle individuals and can also potentially handle the grids that we need to establish the trials. Data collection will have video monitoring, as I mentioned. That will be done in real time and ex situ for review later of each trial to record that feeding and distance covered. 2 phases will be considered, will be used. When we collect our data, we'll collect data in spring, fall for temporal variation. That is also consistent with Stenbers et al. And to measure environmental variables, we will use conductivity, temperature, and depth sensors, salinity sensors, and so forth. You can see a salinity sensor here that's been used previously for this kind of research. Key variables to consider are suspended sedimentation, which is an independent variable in milligrams per liter. Our dependent variables are whether food is consumed and how much, as well as whether movement occurs and what distance may be covered. Food consumption difference between before and after trials will be measured in grams. Distance covered will be measured in centimeters. And body length may be a confounding variable with respect to feeding and movement, so we will also measure that in centimeters. To analyze the statistics, we may have normalization processes such as transformations, log-based transformations and so forth, depending on whether or not the data is normal and how much. Descriptive statistics will be the mean, standard deviation, and variance for that difference in food mass that I mentioned earlier, as well as distance covered. The 4 environmental variables we will measure are salinity, pH, temperature, and hydrostatic pressure, trying to make sure they are within specified ranges so that they can be accounted for as control variables. And the inferential statistics will be t-tests, ANOVA, and possibly a chi-square analysis. The frequentist approach is what I'm advocating for right now because baseline data still needs to be gathered before this research can fully be seen through. We still need to get more information. However, Bayesian approaches may be applicable later. T-tests will help compare the feeding and movement between the control and treatment groups. And ANOVA will be especially helpful when we compare the feeding and movement with the length of sea cucumbers. And some of the literature, as well as the Marine Sanctuary Foundation, noted that sea cucumbers are very slow. So it's possible they do not eat and or do not move in several of the trials, in which case a chi-square analysis would be more applicable to determine more of a yes-no basis and look for relationships in data in that regard. That's it for me. Thank you very much for listening, and I open the floor to questions.